Welcome back to eCoreAcademy, students. My name is Anita Raj, and today, as you can see, I'm bringing you a new biology lesson, and it's going to be our first Biology 1 lesson, also known as Honors Bio. I recently started teaching this course, and in the description box below, you'll see linked my Introduction to Biology 1 video, where I talk all about this course, what we're going to be covering, and honestly, everything that comes with this course, how you can benefit as a student if you take this course at eCoreAcademy. Today, specifically, we'll be kicking off the course with our first lesson. In this lesson, we're going to be understanding components of the scientific method, and as you can see, this is part 1. So this part 1 is going to be, like it says, components of the scientific method. So parts of the scientific method, and honestly, vocab that we need to understand, and we need to understand the process of the scientific method, so that in part 2, I can show you exactly how we implement the scientific method in actual experimentation as biologists. So before I even get into the video today, it's going to be a short one, quick one, hopefully a review from the scientific method principles that you learned in middle school. Before we even get into all of that, I would really appreciate it if you pause this video and quickly hit the like button, the subscribe button, the post notification bell, and even if you could drop a comment down below and share this video once you finish watching it. All of that would be much appreciated. Okay, so let's get right into the first slide. First, I want to describe 2 approaches that scientists take to science in general. So, obviously, science is trying to find reason for natural phenomena that is occurring around us. Back in the Middle Ages and all those time periods, all the way back before the Renaissance, people used religion and concepts that there was a God, that there was this supreme being that controlled what went on. So things that they would witness, such as flood, such as the growth of their crops, such as all these like hurricanes and tsunamis and all of these different things. Those people back then would reason for that with religion, with the fact that there was a God up in heaven controlling all of these natural phenomena that man just cannot control, that man doesn't know where to start reasoning for. So that's where science came about, especially during the Renaissance and Enlightenment periods. So scientists use 2 main approaches to learn about and understand nature and the natural causes of phenomena. So trying to understand the world around us, and nature doesn't necessarily just apply to biotic things like ecosystems. It honestly applies to anything, such as gravity, such as any physics, honestly any type of science. So the process of science applies to any branch of science. We use it to reason for things around us that humans cannot control, but we witness and that are part of our everyday life. There are 2 types of sciences. Those are the 2 approaches that I'm talking about. The first type is called discovery science, and it uses verifiable observations and measurements. So this is where, as a scientist, you're conducting an experiment, you're taking a large volume of data. I want you to emphasize those words. If you're taking notes on this, I would highlight those words and I would emphasize them in your head. Discovery science is using these large volumes of experimental data. So maybe you're doing an experiment on how fast mice can navigate a maze. So the mice and their times, the amount of time it takes for them to get through that maze, that would be your experimental data. That is real stuff that is happening that you're recording so that you can draw conclusions off of that, maybe about mice intelligence and things like that. So you're analyzing a lot of data, a lot of numbers. So you're using verifiable observations and measurements. So you're using a lot of quantitative data, which we're going to be going over in a future slide. So you're using a lot of numbers, a lot of measurements. Maybe you're measuring time, maybe you're measuring volume, maybe you're measuring length. But you're using that kind of data to verify what exactly is going on in that natural phenomenon. The other type of scientific approach that you can take to the world around you is hypothesis-based science, and this honestly piggybacks off of discovery science. So discovery science, you're analyzing large volumes of data to better understand the world around you, whereas in hypothesis-based science, you are taking those large volumes of data and you're trying to form an opinion, a factually correct, a factually supported opinion based off of that large volume of data you analyze. And a hypothesis can be seen as an educated guess. So you're not really sure, but after analyzing all that experimental data, you are pretty sure that is the answer. So this uses data from discovery science to explain science. So like I said with that mice example, you might be recording the time it takes for these different mice to go through a maze. After you collect that data, what do you do with it? That's where hypothesis-based science comes into play. That's where you draw the conclusion. If this happens, then if this happened during the experiment, and this is why it happened. That's pretty much what hypothesis-based science is. For example, let's say the female mice completed the maze faster than male mice. You can say in your hypothesis-based science, using that volume of data that you did collect, you can make a hypothesis that is supported by that data, saying since female mice completed the maze faster, then female mice are more intelligent than males. That is something that would fall under hypothesis-based science. And I hope you noticed that we drew that hypothesis using discovery science, using the large volume of data we accumulated from the experiment. So pretty much in hypothesis-based science, think of it as your opinion. So scientists form their opinions from their experiments. Obviously, these opinions should not be biased so that they're factually correct. You're going to see bias sometimes, but usually the best of scientists will not incorporate bias into their hypotheses. So you're going to see scientists who analyze large volumes of data, but then to try and make a connection between the natural phenomena we're witnessing and the reason for it, they'll draw a hypothesis to reason for that, just like the example I gave you with the mice. And lastly, before we move on to the next slide, I want to go over this kind of flow chart here that I incorporated. I think it's a really great depiction of what the scientific method is. The scientific method is a method we use. It's a process that we use when we start off by witnessing a phenomena and we end that we don't really understand, and we end up trying to draw a conclusion of why it happens. So first you'll see observe. So you're going to make observations. I'm going to talk about on the next slides the types of observations you can make. So let's say with those mice, you might make observations like, for example, you might just go to a zoo or you might go to a pet store and you might see these mice in different intricate like habitats, and you might just observe them and you might see that the female mice move faster and the male mice are a bit slower. So you're making observations. You can say things like, oh, the female mice moved from this end to that end of the pen 1 second faster than her male counterpart did. Then after making those observations, you might develop a question as in, why are the females moving faster than the males? Again, I'm just giving this as an example. I don't know if this is factually correct, but I hope you guys understand what I'm trying to get at by saying these things. Then you're going to form a question, like I said, why do the females move faster? After that, you're going to try to do research to see if you can answer that question based off of experiments that have already been done. So you're trying to find backup research so you're well informed of the topic. Maybe you research how mice anatomy, mice anatomy, mice locomotion, so how they move, and maybe even mice neurology, how their brains work. After that, you might form a hypothesis. You might say something like, I'm totally making this up. Because female mice weigh less than male mice, they're more intelligent and they move faster. You might say something like that as your hypothesis. So your hypothesis might be like, if I put a female mouse and a male mouse in a maze, the female mouse is going to move faster than the male because she has less weight on her. I don't know. I'm just making this up again. Then you would actually conduct your experiment. So you would come up with a procedure, always come up with a procedure, organize the steps that you are going to take, and then execute that. And then after that, based off of the results you collect, you're going to test your hypothesis. So are you going to say, was that hypothesis that I made that female mice move faster because they have less weight on them correct? Lastly, you're going to draw conclusions. You're going to analyze your experiment. You're going to analyze your experiment. You're gonna analyze where you might have gone wrong, where there might have been bias, where you can improve for next time, and then you're gonna finally answer with how and why did your data support your hypothesis. And lastly, you're going to share your results with other scientists who might end up conducting experiments related to this topic in the future. This is a very important slide. So now let's talk about the basis of the scientific method. So what is the scientific method based off of that flowchart that I showed you? So at the end of the day, the observation, the question, and the research are very essential. But what actually starts the experiment? Well, the experiment starts with you forming your own opinion based off of what you've already seen. And that is going to be the hypothesis. So a hypothesis is a proposed explanation for a set of observations, which are questions that can be tested. So a hypothesis can be formed as a question. It can be an if-then statement. You have a lot of flexibility with that. So it is pretty much you looking at a situation like the mice situation I was telling you about, you making observations about it, you asking yourself questions about it, you doing some background research on it, and then finally you coming up with an explanation of why this is occurring, why this natural phenomena is occurring, based off of what you've just seen. You haven't done the experiment yet, you're just basing it off of your background research and what you've seen so far. And a hypothesis has to be the kind of question, the kind of educated guess that can be the basis of your experiment. So it needs to be some kind of question or statement that can be tested and validated and can be accepted or rejected. So before we even get into the hypothesis, what actually makes us think that a natural phenomena that we experience is worth doing research on, worth experimenting about? Well, we observe it, we see it, we witness it, and then we get curious about it. So, what we collect from our senses, what we witness, and why we get intrigued by that is because of the observations we make, the little mental notes that we might make subconsciously saying, hey, this is interesting, this is worth looking into. And there are two types of observations that we make. The first is qualitative, the second is quantitative. And if you don't know what these mean, I'll be showing you on the next slides. So, these sections are going to be called Scientists Make Observations because we're going to be focusing on types of observations. So the first type of observation is a qualitative observation. Like I said, there are two types of observations. Qualitative is the one we're going to be covering first because it's probably, I don't want to see the more basic one, but the one that's a bit broader, it might be easier to understand. So qualitative observation, basic definition I have written here. It is the assignment of value to something that lacks a means of quantitative measurement. So it is pretty much a qualitative observation is pretty much not attaching a number to as a characteristic. So I want you to think of maybe your favorite book character. So whatever book you're reading, maybe it's a fantasy book, maybe it's an autobiography, I don't know. Just think of your favorite book. Think of the main character of that book or your favorite character. So if I was to ask you to name traits that were qualitative traits, you would name things like their hair color, the build of their body, their eye color, their skin color, anything maybe just things like that, maybe the texture of their hair, just basic things like that, the clothes they wear, things that can be described in words. So I could say, my favorite character has black hair. She's tall. She's smart. She has an athletic build. She likes blue clothes. Things like that. That's what qualitative observation is. So with that mice example, qualitative observations are honestly any observation that does not include numbers. Think of it that way. So like it says here, no numbers. So if you thought of the mice, maybe you would describe the color of their coat, the thickness, the coarsity of their coat, just things like that. So you make qualitative observations based off of your five senses. Your five senses are your touch, taste, smell, hearing, sight. Very essential. These are what you base your qualitative observations off of. So if I just looked at something, these are observations I can make just by looking at those things. So if I looked at a mouse, I could just easily say, this mouse has brown fur. This mouse has thick fur by touching the mouse. Things like that. Things that when you look at something, you can immediately tell. And lastly, I want to give you some examples if you're still confused after I gave you all those examples. So if you're doing an experiment maybe about plants, and you're trying to do an experiment where you're trying to water 3 plants and see with different amounts of water, and then determine how the amount of water can impact growth. Maybe that's your experiment. And in that experiment, you could have qualitative observations like the color of a plant. Like if you see here, all these plants are probably the same species, but they're all different colors. So by observing the color of the plant, that's a qualitative observation. By calling this plant here yellow, this plant here blue, this plant here red, those are all qualitative observations. Or maybe the shape of a flower pot that you put the plant in. You could say that this yellow flower pot is put in a cube-shaped flower pot, or maybe this red flower over here is put in like ovular flower pot. Just basic observations like that. So honestly, any observation that does not include numbers or measurements, and which you can easily tell that something like a trait of an object that you can easily derive just by either looking at it, by touching it, by tasting it, by smelling it, or by hearing it using your five senses. So that's enough about qualitative observations. Let's look at quantitative observations. If qualitative observations talk about traits of different components of your experiment that do not include numbers or measurements, then quantitative observations are going to talk about the measurement of a value or quantity using standard measurement methods. So the easiest way to remember this is quantitative. This is the same thing that I've. This is the same memory. This is the same way I've been remembering these two since sixth grade. So quantitative method, in this word quantitative, you have the letter N, and N you can think of as standing for number. So quantitative observations have numbers, qualitative observations do not. That's honestly the simplest way I can put it for you. So the quantitative observations include numbers, like I said. So anything that you can observe by using methods of measurement. So maybe you're taking those mice in that experiment I keep talking about with the mice. Maybe you measure the length of their body using a ruler. Maybe you measure it in inches or centimeters. I don't know. Maybe you put it on a scale, and you measure its mass and its weight. Just things like that. Things that you have to use methods of measurement, and then you collect numbers from that. So examples of this using the plant experiment I was also talking about, you could be measuring the height of a plant. Like if you see in this picture here, there are measurements of the height of the plant, and then also the horizontal, like the length of the plant, if you want to think of it that way, from this frond to this frond. Additionally, you could talk about the pH of the soil. So in this plant experiment I was talking about, we're going to be imagining these plants put in pots, and soil is packed into those pots with the plants. Maybe you'll measure the pH of the soil, and then the value of the pH maybe it's 7, maybe it's neutral. That would be a quantitative observation because that's a number, and you used a pH strip, which could be seen as a standard of measurement to find out what the pH of the soil is. So those are examples of quantitative observations. Next, let's look at what inferences are. So inference is an educated guess based on observations and data. So it could be pretty much I don't want to say it's the same thing as a hypothesis. Those two are definitely related. You can think of them kind of as synonyms, but for now I want you all to think of inferences as their own idea. So inferences are an educated guess based on observations and data, just like I said. So you have all these qualitative and quantitative observations, and you have data that you probably collected from your experiment. For example, that mice experiment at how fast each mouse was able to complete the maze in. You would take those volumes of data and then your observations, qualitative and quantitative observations about your test subjects, and you would analyze them. And based off of those, you would make an educated guess, and you try to connect what you've seen in your experiment to natural phenomena you also witness. So here's a great example, a very simple example actually of an inference. So here in this picture, I just want you to take a minute to look at it. It's of a child because we see a child's legs, and this child's wearing rain boots, and this looks like a rain jacket, and the child is standing in a puddle. You can see that it's a puddle because obviously you see ripples on the surface. There are leaves floating on the puddle. So based off of this picture, what inference can we draw? Because we're not given a lot of information, but we can, just by looking at this scenario, we can come up with an educated guess of what's going on. So we start off by identifying exactly what's going on in the picture. So in one sentence, the child is standing in a puddle, as you can see. And then after we make this observation, after we make this observation that the child is standing in a puddle, this is where the inference part of it comes in. So we're reasoning, why is the child standing in a puddle? Why is there a puddle? First off, why is there a puddle? So that's where we draw an educated guess based off of what we've seen and based off of the natural phenomena that we understand. So it may have rained recently. We all know that when it rains, in ditches and places that dip down a bit in the ground, water can collect for a bit of time, especially if it rained hard. And if it rained recently, that water is going to sit there, as opposed to having been like a month since it rained. That water is obviously not going to be sitting there for a month. So obviously, if water is collected there, we immediately infer that it must have rained recently, and that's why the child is going outside to probably play in the rain. So that's what an inference exactly is, and I think this example here is key to your understanding what an inference is. Next, I want to talk about variables. So you probably know about variables from math. When you were in sixth grade, your teacher was probably saying something like, "A variable is a letter from the alphabet that represents an unknown variable in algebra." I'm just telling you exactly what my sixth grade teacher said to us when we were like, "Huh, what's a variable?" in sixth grade. But in science, a variable retains that same meaning, but it's a little bit different. And here's the exact definition of what a variable is. A variable is any factor, trait, or condition that can exist in different amounts or types. So a variable is any factor, trait, or condition that is unknown, that we really don't know. And we don't know that until its amount or type is specified. So it's a variable. Think of the word vary, so it can change, and the nature of a variable will change depending on the experiment and the scientist who is conducting that experiment. There are two main types of variables that you will encounter in science, especially when you're doing experiments. The first type of variable you're going to see is an independent variable, and you usually abbreviate this as IV, like an IV drip. This variable that is the one that is purposely changed or manipulated. So in a scientific experiment, this is what the scientist is purposely changing. So this would be like maybe the difference between the different test subjects. So if you're thinking about a plant experiment like I was talking about, where we have three flower pots and each one has a different plant, and we're trying to see how if we water one plant, let's say with 1 liter of water every day, one plant with half a liter of water every day, and one plant with no water at all. We're trying to see how that affects the height of the plant. I'm just giving you an example of an experiment. In this experiment, the independent variable would be the thing that we as the experimenter are changing. Again, I want to say that the independent variable is what we are changing. It is what we are in control of. It's what we're purposefully manipulating. And in this case, we're controlling the amount of water that each of the plants get. As the person who's doing the experiment, if I was the person doing the experiment, I would be the person deciding which plant gets 1 liter, which plant gets half a liter, which plant gets 0 liters. I would be the person deciding how much water each plant gets. So that's what I'm manipulating. I'm manipulating the amount of water. So the amount of liters of water each plant is getting. I want to segue into the dependent variable, which is the other variable you'll have. So this is known as the responding variable because it changes in response to the manipulated variable. So because I'm changing the amount of water each plant gets, the dependent variable is going to be what occurs because I'm giving those plants these specific amounts of water. So it changes in response to the manipulated variable. I'm controlling the manipulated variable, but keep in mind that I cannot control the dependent variable. The dependent variable is going to act depending on how it should based off of what the independent variable is. So let's say I'm giving plant A 1 liter of water, I'm giving plant B 0.0 half a liter of water, and then plant C no water at all. And let's say 0.5 liters is the appropriate amount of water I should be giving a plant. That's going to be resulting in optimal plant growth. Let's just say that. But because I'm doing this experiment, I don't know for sure. So I don't know that while I'm doing this experiment. And let's say by the end of the experiment, plant A, which I was giving 1 liter to, it's a decent height, but it's kind of drooping a little bit. I'm just making this up. Plant B, let's say, is the tallest plant, the healthiest looking plant, and let's say plant C is dead, shriveled. It's collapsed. There's literally nothing left of it. And what at the end of that experiment, let's say to determine optimal plant growth, which amount of water gave each plant the best opportunity to grow, I end up measuring the height of the plant. Let's say that is going to be my dependent variable, and I measure the heights of each of the plants. Think of it this way: the dependent variable is going to be the height of the plant. That's because I can't control the height of the plant. I can control how much water each plant gets, but by the end of the experiment, I can't control how much each plant has grown because the amount of growth is going to be in response to the amount of water I give. And I can't control that. So think of it this way: independent variable can control, dependent variable respond to the independent variable, but you can't control it. And usually the dependent variable is going to be some kind of measurement that you take, like the height of the plant or the pH of the soil, or I don't know, the width of the leaves, things like that. Next, I want to talk about control. So in this case, when you think of control, I want you to think of a controlling person. So maybe you have met someone who you consider controlling, someone who wants everything to go their way and will make sure that you act a certain way depending on what they want you to do. In layman's terms, you can think of someone controlling as someone who changes standards to meet what they want and then doesn't want those standards to change. So they might be very resistant to change. That is exactly what a control is in a scientific experiment. And the control is going to be something that we establish that we don't change. And the reason we have a control in experiments is because it's a standard for comparison that is used. So let's say at the bottom of this page right here, this is a perfect example. Let's say plant A here is going to be the plant I give half a liter of water to. Let's say this middle plant is going to be the plant I give 1 liter of water to. And this plant over here is going to be the plant I give no water to. So this plant over to the right, the one I'm giving half a liter to, maybe I started the experiment knowing that the optimal amount of water I should be giving a plant is half a liter. So I'm purposely giving this plant half a liter because it might be like the average amount of water people give that plant, something like that. So this would be like my control. This would be the thing that is standard, that is average, that is normal. And these two over here, the plant that's receiving no water at all and the plant that's receiving a lot of water, which is 1 liter of water, these are going to be my experimental groups. So this plant over here on the right, which is the half liter, it's going to be what I use to compare. So at the end of the experiment, if any of these plants are taller, then I can say that more water than what is normal, than what is what was given to the control, results in optimal plant growth. And maybe if the one on the left is shorter, maybe shriveled and dead because I didn't give it any water, I can compare its height to the control and say giving less than 0.5 liters of water will result in plant death. And I'm using the standard of comparison I'm comparing both these experimental groups back to this comparison so that then I can compare those experimental groups together. So the comparison between the experimental groups in response to the manipulated variable. So the manipulated variable is again the independent variable. We're just trying to compare how different plants in this case are responding to different amounts of water. And we're doing that by measuring the dependent variable. Like I touched on, the control versus the experimental sample. So this is what is different. The control is not treated as it's not exposed to the experimental factor. In this case, you can think of it like plant A over here is not exposed to abnormal amounts of water. It's given the standard 0.5 liters. However, the experimental is treated. So it is given that it is going to be exposed to the experimental factor, which in this case are the abnormal amounts of water. So if you want to think of it treated, you can think of patients. So you could have like patients, and then you might have like a cure for a disease or something. I'm just coming up with this, but think of it as treated as this patient is being given the cure. This patient is being used as a standard of comparison, is not being given that cure or that medicine for this sake. So treated control, not treated with the experimental factor, not treated with that medicine or whatever it is, and the experimental factor is being treated with that medicine or however you want to think of it. So that's all I have for you today. I hope that was a good review for many of you who might have forgotten these concepts from middle school that you learned about the process of science. Again, this is where I show you the components of the scientific method, like those vocabulary terms and examples and all those things. And then in the next video, I'm going to be showing you how we use the scientific method in actual experimentation, and I'll be showing you how to develop simple experiments that you'll be able to carry that method through biology, chemistry, physics, anatomy and physiology, honestly any science course you do, and it'll help you stay organized during those procedures and come up with the best results and come up with the best conclusions and reports. Like I said, that's all I have for you today. If you enjoyed this video, please hit the like button, subscribe button, and hit the post notification bell. Drop a comment to let me know what you think. I'm starting this biology course, and I could really use feedback from all of you about things you want me to incorporate, things you like, things you want me to change. Honestly, anything. Just let us know. Lastly, we would appreciate it if you use Facebook, Twitter, and Instagram, our social media platforms, to share this video and show your support. We also have a Gmail, so if you have any personal, business correspondences, honestly anything, feel free to reach out. We check that religiously. It's eCoreAcademy@gmail.com. You can find it in the description box below as well. Lastly, we have our website that's gone up. Links also in this description box. On there, you'll get a bunch of cool features like notes and quizzes and all these lessons organized into easy into more I want to say student friendly into a more student friendly format for you guys to access. Like I said, that's all I have for you today. Hope you enjoyed the video. Let me know what you think about this new Biology 1 course, and see you in the next one. Bye.